Perfect. So I'd like to thank again Hunter's Hope for providing the opportunity for me to review therapeutics or treatment for CTX today as well. So I'm going to show the bile acid pathway in CTX again. And as I mentioned before, the deficiency or the defect in CTX is a deficiency in CYP27A1 or the sterol 27 hydroxylase enzyme that results in a deficiency of chenodeoxycholic acid and the cholic acid, the primary bile acids. And as I mentioned, in particular, CDCA normally acts as a ligand to activate the FXR and provides an inhibitory cascade resulting in loss of activation of CYP7A1, that initial step in the 7-alpha hydroxylation of cholesterol. And the lack of CDCA in particular results in kind of highly induced activity of CYP7A1, which results in accumulation, in particular, of cholestanol and bile alcohols that are present in the blood and urine in CTX. And these are derived from intermediates in the bile acid pathway, 7-alpha hydroxy-4-cholesterol-3-one or 7-alpha C4, which is 12 hydroxylated to form 7-alpha 12-alpha dihydroxy-4-cholesterol-3-one or 7-alpha 12-alpha C4, which generates these bile alcohols, triols, bile tetrols, bile pentols, and bile hexols, which are quite particular to CTX. And cholestanol is thought to be formed in a pathway from 7-alpha C4. An accumulation of these intermediates is thought to result in the signs and symptoms of CTX. And so for the last 40 years or so, replenishment of the chenodeoxycholic acid that's missing in CTX has been used as a treatment in CTX. And replenishing the chenodeoxycholic acid is 3 pills a day, very simple treatment. Results in it lowers the activity of CYP7A1 and lowers accumulation of the bile acid precursors and the derived cholestanol, bile and bile alcohols. So the first evidence that CDCA was a treatment in CTX was back in the early 80s and was published by Jerry Salen, where he treated CTX patients with 750 milligrams of CDCA per day and showed that the plasma 5-alpha cholestanol was decreased, and that in these patients, they showed improved EEG abnormalities. And then in a landmark study in 1984, Bergener, Salen, and Schaefer treated 17 CTX patients with CDCA over 1 year. And they showed a decrease in the elevation of cholestanol and bile alcohols. And they showed that the neurological deterioration was stabilized in the disease and that disease progression was halted. And so since that time, treatment with CDCA has been the standard of care in this disease. CDCA has been globally recognized as the standard of care. CDCA dosing in adults, as I said, was 750 milligrams. That's 3 250 mg tablets per day. And in children, the use of up to 15 mg per kg per day. So dose adjustments are made based on body weight, the clinical signs and symptoms, monitoring of those, and monitoring the cholestanol response to treatment, as well as monitoring liver enzymes to rule out the very low risk of hepatotoxicity from CDCA. It's been shown in 2 large cohort studies that early treatment before the age of 25 provides improved patient outcomes. And it's been shown in other studies that treatment cannot reverse the neurological damage that's already occurred in the progression of the disease. So although there's no specific guideline available for the treatment of infants diagnosed through newborn screening, there are expert consensus guidelines available for the diagnosis, treatment, and management of patients with CTX of all ages that were generated around 5 years ago using a modified Delphi method. So in that survey, all expert panelists agreed that CDCA is the lifetime replacement therapy for CTX. And all panelists agreed first that the most beneficial time to start CTX treatment is from birth following a positive newborn screening test. So in accordance with the results of the Delphi study, a clinical specialist managing a neonate diagnosed with CTX would initiate treatment from shortly after birth, ideally within the first months of life. So I wanted to talk a little bit about the regulatory status for CDCA. So in Europe, based on the data from two retrospective cohort studies conducted in CTX patients who underwent CDCA treatment, in these studies, eligible patients were aged between 2 and 75 years, had been diagnosed with CTX, and were treated with CTX orally for more than 1 year. This is the publication here, the safety and effectiveness of CDCA treatment in patients with CTX. So the impact of CDCA treatment on biochemical markers and disease signs and symptoms were assessed in addition to the safety and tolerability of CDCA treatment. So signs and symptoms of disease resolved, improved, or remained stable in many patients with concomitant improvements in biochemical marker levels. So serum cholestanol statistically significantly decreased, as well as 7-alpha-hydroxy-4-cholestin-3-one, or the 7-alpha-C4 molecule. And so based on the evidence from these two cohort studies, EMA provided approval for chenodeoxycholic acid Ledian in 2014. They designated this an orphan medicine. And in 2017, CDCA was authorized to treat adults and children from 1 month of age with CTX. And again, the dose in adults was 750 mg, 250 mg 3 times a day, adjusted depending on the patient's age, liver function, and levels of bile acids in their blood and urine. And they note that for small children, the contents of capsules can be mixed with sodium bicarbonate solution to make a liquid. So in the US, the situation was different. Although since the trial in the beginning of the 1980s, CDCA was the standard of care, it was used off-label for CTX. So in 1983, the FDA did approve CDCA for the treatment of gallstones, but they did recognize that CDCA was a medical necessity for patients with CTX. And in 2009, they did designate CDCA an orphan medicine. And this was in large part to patient advocacy efforts through the United Leukodystrophy Foundation. And, you know, before this time, patients weren't actually, CDCA wasn't being marketed in the US anymore. They had to ship the medication in from Europe. And so at that point, Manchester Pharmaceuticals started marketing CDCA in the US, and it became available again. And although this occurred, the FDA was not willing to approve CDCA for the treatment of CTX until a randomized double-blind controlled study was performed. And as you can imagine, this was a very challenging study to perform because CDCA was the standard of care. It's been shown to be an effective treatment, and patients and physicians didn't want to come off the treatment to perform this study. But a study was able to be performed, and the efficacy of CDCA for the treatment of patients with CTX was evaluated in a prospective, double-blind, placebo-controlled, randomized crossover withdrawal trial, which was the RESTORE study. This 24-week trial demonstrated that treatment with CDCA at 250 mg 3 times per day resulted in significant reduction in plasma cholestanol and urine 23S-pentol, the bile pentol, compared to placebo treatment. And based on the results of the RESTORE study, back earlier this year in February, the FDA approved CDCA finally for the treatment of CTX. And so these are the results of the RESTORE study, which were just published actually last week in Genetics in Medicine. And again, the FDA approved based on the data from this clinical trial, phase 3 clinical trial, evaluating the safety and efficacy of CDCA in adult patients with CTX, with the primary endpoint being measurement of the urine bile alcohol, 23S-pentol, and in other additional secondary measures. So the biomarker generated for the primary endpoint of reduction in urine 23S-pentyl was highly statistically significant at the end of the randomized double-blind withdrawal periods. So you can see on the right-hand side here, let me see if I can use the mouse. At the top right, there are the grey lines are the randomized withdrawal periods, and the participants were split into two groups. One is the red and one is the blue. And the red group had the withdrawal for the first period. And you can see on withdrawal of CDCA, the cholestanol, which is the top graph, increases. The next graph down is 7-alpha-C4. And so you can see a very great increase in 7-alpha-C4 in the red group on the first withdrawal. And the bottom graph is the urine bile pentyl, the 23S-pentyl. And you can see the same increase in 23S-pentyl. And then the blue group is the second withdrawal within the second grey areas. And you can see again an increase in all of those. And other biomarkers were measured as secondary endpoints in the trial. And so I just show here the graphs for 7-alpha-C4, which is that first rate limiting step of the 7-alpha hydroxylation, cholestanol, and the bile pentyl that's excreted at high levels in the urine. So there is an alternative to treatment with CDCA, which is treatment with cholic acid. So cholic acid is a less hepatotoxic primary bile acid. And so in infants, because of the lack of pediatric approval by the FDA, cholic acid treatment is also a viable treatment option for infants with CTX. And it can typically be used when CDCA is no longer tolerated, for example, through hepatotoxicity or effective. Cholic acid was approved by the FDA in 2015 for the treatment of pediatric and adult patients with bile acid synthesis disorders due to single enzyme defects, and this includes CTX. Cholic acid should be dosed according to a patient's weight. The recommended dosage is 10 to 15 mg per kg taken once per day or in 2 divided doses in pediatric and adult patients. Although cholic acid is approved for the treatment of CTX, long-term treatment data in CTX are lacking, and it's generally considered a second-line treatment when CDCA-induced hepatotoxicity occurs, or as a first-line treatment in affected infant with cholestasis. In terms of EMA, in 2009, cholic acid received an orphan drug designation. In 2015, it was approved for the treatment of inborn errors in primary bile acid synthesis in adults and children from 1 month of age. It should be noted in Europe that in 2020, the marketing authorization was withdrawn, not because there was any concern about the drug, but the sponsor decided to permanently discontinue the marketing of the product for commercial reasons. There is a alternative cholic acid called Orfacol, which can be used. I'm just going to go through this, which again is approved for pediatric and adult use. So in conclusion, treatment with CDCA has been shown to be safe and efficacious in infants with careful monitoring, with dose adjustment is needed, and can be titrated upwards to a maximum dosage of 15 mg per kg per day in older children to maintain adequate metabolic control. And the expert consensus is that treatments can be effectively managed with a minimal risk of adverse outcomes. I just wanted to, one last sentence, say that although there's no specific treatment guideline available for infants diagnosed through newborn screening, there are these Delphi method guidelines that were generated 5 years ago. Hunter's Hope is now helping us develop specific treatment guidelines for infants diagnosed through newborn screening. So hopefully this is something in the pipeline that will be helpful. And I'll finish up there. That's it.